My name is Christopher Fry. I'm a Director of Epigenetics Product Development at Cell Signaling Technology, and today I want to talk about the ChIP assay and really talk about things to consider when you're planning a successful ChIP assay. So I'll spend just a couple minutes briefly giving an introduction to ChIP, both ChIP and ChIP-seq, and then I want to talk about really three keys to a successful ChIP experiment. The first one being an optimized protocol, high-quality reagents. Next, we'll talk about crosslinking and chromatin fragmentation. And then finally, we'll talk about ChIP-validated antibodies. So what is ChIP? ChIP is a powerful technique that's used to identify and analyze protein-DNA interactions within the genome in vivo. You can use it to look at binding of histones and histone modifications, transcription factors and cofactor proteins, replication factors and DNA repair proteins. And you can use this method to look at single gene analysis using ChIP-qPCR. Here is where you would look at a handful of interesting target genes. Or you could use it and look at whole genome mapping of the protein or histone modification. Now in the ChIP assay, cells or tissues are first fixed with formaldehyde, and this cross-links histones and non-histone proteins to the DNA. The cells or tissue are then lysed and nuclei are prepared. Chromatin is fragmented either by sonication or enzymatic digestion into 150 to 900 base pair DNA protein fragments. Once that is done, the nuclei are lysed and fragmented and chromatin is isolated. We then can add antibodies that are specific to histones or non-histone proteins, and then we add protein G or protein A beads to help IP and enrich these antibody chromatin fragments from the chromatin sample. We can reverse the crosslinks and purify the DNA, and then we can analyze this DNA to see what DNA we've immunoenriched using the antibody. And this can be analyzed by one or two methods, ChIP-qPCR or ChIP-seq. With ChIP-qPCR, you can design primers against a handful or so of target genes of interest and use qPCR to identify and quantify the enrichment of these target genes. With ChIP-seq, you can actually generate a DNA library and then identify and quantify and map these binding reagents back to the whole genome. Next, I wanted to spend most of my talk really focusing on three areas that to think about when planning a successful ChIP experiment. The first one is use of reliable, optimized protocol. Next, we'll talk about cross-linking and fragmenting chromatin. And finally, we'll talk about the antibodies that you use in your experiment. So when starting a ChIP protocol, you really want to use a reliable, optimized protocol, whether it be a homebrew protocol that you find that is well published or is being used by a ChIP lab, or it's a kit that you buy from a company. And at CST, we provide multiple chromatin IP kits. Here's two on the slide here: the Simple ChIP Plus Sonication Chromatin IP kit and the Simple ChIP Plus Enzymatic Chromatin IP kit. Both kits use magnetic protein G beads. They work with cells and whole tissue samples. They're compatible with ChIP-qPCR and ChIP-seq, and they work for histones, transcription factors, and cofactors. So really, these kits both work for really, everything. You don't need a specific kit for specific protein types. These kits contain all the buffers and reagents required to do the ChIP. They have a complete detailed protocol. They're actually simpler than most competitor kits. They have DNA purification columns, which are required for downstream qPCR and NG-seq analysis. And they also contain a universal positive control histone H3 antibody and negative control normal IgG antibody. And using these antibodies in combination with the positive control primer sets really helps ensure your experimental success, regardless of whether your antibody of interest is working or not. These antibodies will tell you that your chromatin is working for an IP experiment. So next, I want to spend some time talking about cross-linking and fragmenting the chromatin. So there are two methods that can be used to fragment chromatin: enzymatic digestion, shown here on the left in blue, and sonication, shown here on the right in orange. So with enzymatic, cells are lysed in a low detergent buffer. We then fragment by digesting with micrococcal nuclease. And micrococcal nuclease is an enzyme that digests double-stranded DNA in between the nucleosomes. Once the digestion is complete, we briefly sonicate to lyse the nuclei, and the chromatin is purified by centrifuging out the insoluble material. Now, MNase digestion is very mild. It's done in a low detergent buffer at 37 degrees, and we actually recommend doing a partial digest. And what you get is shown here in the gel electrophoresis on the right. The second lane shows DNA that is purified from a MNase-based chromatin digestion. As you see here, you see a ladder of DNA fragments from 150 to 700 base pairs. So in orange on the right is shown the sonication protocol. Here we do a cell lysis in a low detergent buffer, and then we sonicate the chromatin, and we use acoustic energy to sonicate the chromatin into small fragments. Now, the sonication can be done in a number of different conditions. Some protocols use a low detergent buffer. Other protocols use a high detergent buffer. And different types of sonicators can be used. Traditionally, a probe sonicator has been used for ChIP, but newer water bath and ultrafocus sonicators can be used. Regardless of the sonicator used, you need to do a bit of optimization for the sonication protocol. And this is important because there's different classes of ChIP targets there are different classes of protein targets that behave differently in the ChIP assay. So there's histones, which are very stably bound to DNA, and there's a large number of histones per DNA fragment. These proteins are thus relatively easy to ChIP, and they're insensitive to harsh conditions such as high detergent and heat that's created in the sonication protocol. DNA-binding transcription factors are less stably bound to the DNA. There's fewer factors bound to the DNA fragment, and these are more difficult to ChIP. They're much more sensitive to harsh conditions. And finally, transcription cofactors, like the switch-SNF complex proteins, often do not directly contact DNA. They're often part of these large protein complexes. The antibody epitopes might be partially buried, and these are actually very difficult to ChIP and very sensitive to harsh denaturing conditions. Now, again, with sonication, one wants to do a bit of optimization with the protocol. And one of the important things to consider is the number of cycles of sonication or sonication time you're using in your assay. Shown on the left here is a gel electrophoresis of DNA fragments that were generated by sonication. And on the bottom, we're looking at increasing time of sonication, as shown here. As you can see, as we increase the time of sonication, we see smaller and smaller fragments of DNA. And looking at this, one might think that 8 minutes of sonication might be the best because it gives us a good, nice, tight band of chromatin fragments. However, if we look at enrichment of target genes for the TCF4 transcription factor and beta-catenin, a cofactor that binds to TCF4, we see that the highest levels of enrichment for CAM-K2D, a known target gene, are found using chromatins that was sonicated for only 2 to 4 minutes. We find that 8 to 10 minutes actually decreases the enrichment that we see by qPCR. So more sonication is not always better. And so what you want to do is you want to use the minimal number of cycles that you can to get chromatin around the size of 100 to 500 or 100 to 700 base pairs in length. And that should allow, if you're using the minimal number of cycles, should give you the best enrichment. Now, another thing to consider is the amount of detergent in your sonication buffer. Even with optimized cycles, if you have too much detergent, you can lose signal for some target protein types. Now, histones are very resistant to high detergent levels. As shown in this slide, we're looking at enrichment of trimethylk27 across a region of the genome. Here we're using our Simple ChIP enzymatic chromatin IP kit, our Simple ChIP sonication chromatin IP kit, which has a low detergent buffer, and a leading competitor sonication kit that contains a high detergent buffer. If we look at enrichment of trimethylation 27, you see that all 3 of these kits work equally well, giving us nice enrichment that we would expect to see in this region of the genome. However, if we look at transcription factors and cofactors, we see a big difference. Here we're looking at TCF4 and 2 of its cofactors that it recruits to its target genes, beta-catenin, and CBP. If we look at the ChIP-seq data, here we're looking at the ACSL5 target gene. We see nice enrichment of these 3 proteins using Simple ChIP enzymatic kit, the Simple ChIP Sonication kit, but using the high detergent competitor Sonication kit, we see absolutely no enrichment. And this is again because the high levels of a detergent are actually denaturing the chromatin and disrupting the antibody epitopes. And so when you're looking at transcription factors and cofactors, you really want to use either enzymatic or a low detergent Sonication buffer. What about tissue ChIP? Which method works best for tissue ChIP? Well, we found that both methods, the Sonication and the enzymatic, work equally well in most tissues. So shown here is ChIP-seq data using mouse liver, and here we're using our Simple ChIP Plus Sonication chromatin IP kit on the left and the Simple ChIP Plus enzymatic kit on the right. In the top 2 panels, we're looking at enrichment of trimethylate lysine 4 and the FOXA1 transcription factor. And here we see very similar enrichment patterns using both enzymatic and Sonication, as we would expect. At the bottom, we're looking at enrichment of ring 1B and SUS12 here at the HOXA loci. And again, we see very nice enrichment of both polycomb responsive complex 1 and 2 containing these factors at this loci. So with enzymatic ChIP, it's really important with tissues to disaggregate your tissues into single cell suspension before you do the digest. And this can be done either using a tissue disaggregator like the Becton Dickinson Medi machine, or by using a Dounce homogenizer. But once you get that single cell suspension, it's very easy to do the digest. With Sonication, the Sonication itself will actually disaggregate the tissue while it's fragmenting the chromatin. However, again, with Sonication, we find that we have to do some additional optimization to get efficient enrichment of transcription factors and cofactors. So what we find with tissues in Sonication, that increasing the cross-linking time helps out a lot when trying to ChIP for transcription factors and cofactors. So shown on the left is a gel electrophoresis of DNA fragments that we've prepared by Sonication for mouse heart, mouse brain, and mouse lung. And you can see with increasing cross-linking, we do get a trade-off and we get larger chromatin fragments. However, if we look at enrichment of HOXD10 and HOXA1 by ChIP-qPCR, we see that ring 1B and SUS12, both of which bind this region, show increased enrichment when we cross-link for 30 minutes versus cross-linking for 10 minutes. And we see absolutely no increase in enrichment of this GAPDH gene, which is it's not a target of these complexes. And so we find again, with Sonication, if we cross-link just a little bit longer, we can actually increase our enrichment. All right, so to just summarize the first part of my talk, tips for cross-linking and chromatin fragmentation, I'd like to remind you that I showed you that histones are resistant to harsh fragmentation conditions like heat, detergent, and the protein denaturation that's caused. Transcription factors and cofactors are sensitive to these harsh conditions. The enzymatic-based fragmentation is mild, and it works well for histones, transcription factors, and cofactors. Sonication does too, but you have to be you have to do some optimization and keep some things in mind. And that is, you want to use mild buffers that have low detergent. You want to keep your samples cold. You want to minimize your Sonication cycles to really balance the chromatin fragment size and the enrichment levels of your target of interest. And finally, for tissue, you may want to increase the cross-linking time to maximize your transcription cofactor and transcription factor ChIP enrichment. For more information about ChIP, you can actually download our guide for ChIP and ChIP-seq at www.cellsignal.com/ChIP. Okay, so I want to spend the last part of my talk talking about antibodies. Antibodies are also very important for the ChIP assay. And so for the ChIP assay, our group validates our ChIP antibodies using relevant model systems. And I just show an example here on this slide. Here we're looking at validation of the glucocorticoid receptor rabbit monoclonal antibody. Here we're testing this antibody on A549 cells that are either untreated or treated with dexamethasone. Now we know that treatment with dexamethasone induces nuclear localization of GR and binding of GR to its target genes. In this experiment, we're looking at binding to SLC19A2 and MT2A in both untreated and treated cells. And you see very nicely, we see enrichment only in the treated cells as expected. We also see no binding to this alpha satellite repeat element, which is our negative control. So here the antibody is showing exactly what we would expect to see, and that is enrichment of GR targets in treated cells. Now when testing an antibody in your ChIP assay, I always advise to titrate the antibody. This is something we do for every antibody that we test. Shown here is a titration for our EZH2 rabbit monoclonal antibody. And here, what you're looking at is looking at enrichment of the HOXA1 and HOXA2 loci as we increase concentration of the antibody in the IP assay. And you see, as we increase the amount of antibody, we do see an increase in enrichment. However, that enrichment levels off and then actually falls with increasing amounts of antibody. And so what we find with this antibody is that 2.5 to 5 microliters of antibody per IP is ideal in generating the best signal to noise, the best enrichment levels of these target genes. So with an antibody in ChIP, more is not always better. I know some people still believe that you need to use 5 or 10 micrograms of antibody for a ChIP assay, and that's simply not true. Sometimes as little as a half a microgram actually works best. And we find that it's the ratio of the antibody to the chromatin that's important. So we always titrate the antibody. Again, when selecting your antibody for ChIP, it's always good to look at validation data across different applications, as this often tells you more about the antibody's specificity and sensitivity. So here's an antibody that we have against BRG1. Again, it's a rabbit monoclonal antibody. In ChIP-qPCR, shown in the upper right, we're looking at enrichment of actually 2 estrogen receptor target genes. Estrogen receptor recruits the BRG1 switch NIF complex to target genes when cells are treated with estrogen. So here we're looking at MCF7 cells treated with estrogen. And as expected, you see nice recruitment and enrichment of BRG1 to these target genes. Shown below is ChIP-seq track, again looking at enrichment of another target gene, ENZA. And again, we see very nice binding and enrichment of BRG1 at the ER binding site in this gene. On the left is shown a Western blot. Here we're actually looking at expression of BRG1 across a variety of different cell lines. These are whole cell lysates, and as you can see, this antibody really detects only a single band in these whole cell lysates. Now with BRG1, we know from mRNA expression data that A549 cells do not express BRG1. As Segal pointed out, BRM is a paralog of BRG1 that's actually highly homologous in sequence to BRG1, and BRM is expressed in these A549 cells. So we know since our BRG1 antibody does not detect a band in these cells that it's not cross-reacting with BRM, and so we know this antibody is specific to BRG1. With glucocorticoid receptor, we already went over the ChIP-qPCR data, or the ChIP-qPCR data. Shown below is the ChIP-seq data, and again, looking at a known target gene, we see a single peak of enrichment here over a GR binding site. Very good signal-to-noise ratio. Again, looking at specificity and performance of the antibody across other applications, if we look at IF, like here we're looking at A549 cells, again, that are untreated and treated. And the left, we're looking at untreated, and as expected, you see both nuclear and cytoplasmic localization. Once we treat the cells, we see just nuclear localization, as expected, since dexamethasone induces nuclear translocation of the protein. And finally, we have a Western blot on the left. Here we're looking at whole cell lysates from 293 cells that are either untransfected in lane 2, transfected with an overexpression construct for MYC-tagged GR in lane 3, or transfected with a construct overexpressing MYC-tagged mineralocorticoid receptor in lane 4. And again, we see this antibody is very good at detecting endogenous GR, overexpressed GR, but it doesn't cross-react with their mineralocorticoid receptor protein. again showing that it's specific for GR. So I want to spend a little bit of time talking about trimethyl H3 lysine 27. Again, here we're looking at a Western blot of whole cell lysates, and the antibody detects a single band that corresponds to histone H3. In IF, we see the expected nuclear localization, and in ChIP-qPCR, we actually look at enrichment of known target genes MyoD1 and MYT1. These are two genes that are repressed in these HeLa cells, and they're repressed by polycomb and repressed by this trimethyl H3 lysine 27 mark. And we see no enrichment of GAPDH or RPL30, which are housekeeping genes that are active. The ChIP-seq data is shown below, and we see the expected kind of broad binding of, or broad localization of this mark across MYT1 as expected, as this is what this is how polycomb works on its target genes. So by all accounts and different applications, this antibody looks very specific. However, we know with the histone modification antibodies that histones themselves are highly modified on lysines, both by methylation and acetylation, and these antibodies have the potential to cross-react with other methylated lysines. And so for all of our methyl lysine, acetylysine, and methyl arginine antibodies, we do a histone peptide array analysis where we titrate our antibodies on an array of peptides that contain all the known modifications of histone H3, H4, H2A, H2B, and histone H1. And what we look at is we look at peptides that have a single mark and peptides that have dual marks containing neighboring modifications, so we can look at the effect of neighboring modifications on binding of the antibody. So for this trimethylation 27 antibody, we see in column L that the antibody binds very nicely to a peptide containing trimethyl H3 lysine 27. However, we see no binding in K and J, which correspond to dimethyl and monomethyl, and no binding in H, which corresponds to trimethyl H3 lysine 9, which is highly similar in sequence to the trimethyl lysine 27. So with this antibody, again, we see no binding across peptides containing a single histone methylation site. And then over here on the right, if we look at dual sites, we see that the antibody can bind when an upstream arginine 26 is also dimethylated, again, showing that this antibody doesn't care whether the upstream arginine is methylated. So this antibody is highly specific to K27 trimethyl. So in my last two slides, I just want to talk about the performance of rabbit monoclonal antibodies in the ChIP and ChIP-seq assay. I know there a lot of people believe that polyclonal antibodies work better for IP-based experiments, but that's simply not true. Some of our best ChIP and ChIP-seq antibodies are rabbit monoclonal antibodies. Shown here is a comparison of our trimethylation 27 antibody against an code-approved competitor polyclonal antibody. And we're looking at enrichment of HOX-D and MyoD loci at the top and at the bottom. And what we find is with the CST antibody, we see actually better levels of enrichment and better signal to noise than we do with the ENCODE-approved competitor polyclonal antibody. The next slide shows a rabbit mono that we have against RNA polymerase, largest subunit, RPB1, the N-terminal domain. This antibody is used to look at total RNA polymerase binding across the genome, as the N-terminal domain does not contain a lot of the phosphorylation modifications that the C-terminus contains. And so again, we're looking at enrichment of RNA polymerase using our RPB1 rabbit monoclonal antibody and the competitor polyclonal antibody. This is a leading polyclonal antibody that is used quite often for ChIP and ChIP-seq. And we're looking at enrichment of the ZNF740 and the beta-actin genes. And again, what we find is the rabbit monoclonal antibody is superior and actually gives higher levels of enrichment and better signal to noise than the polyclonal antibody. And as expected, you see high levels of enrichment over the promoters of these genes, and then you see lower levels across the body of these genes. So finally, I'd like to summarize the last part of my talk, talking about the use of ChIP-validated antibodies in the assay. So when using an antibody, always use a ChIP-validated antibody if possible. But look at the data across other applications to look at specificity and sensitivity. Use the antibody at recommended dilutions if it's already provided. If not available, try using IP-validated antibodies, but optimize for ChIP by titration. And again, rabbit monoclonal antibodies work very well in ChIP and ChIP-seq. At CST, we have over 280 ChIP-validated antibodies, and over 50 of those are ChIP-seq validated rabbit monoclonal antibodies. For more information, you can visit our website at www.cellsignal.com/epigenetics or /ChIP, and that'll provide a lot of information about our antibodies, our ChIP kits, and epigenetics in general. Thank you.